Recording in
I'm so sorry, but the quality and sound of the original recording didn't turn out too well, so I'm re-recording today's lesson right now. So that's why there aren't any interactions in this recording and why the recording is a little shorter. But anyways, I'm Alice. Welcome to week 1 of the Bio and Med Bootcamp. Thank you so much for taking the time to join from wherever you are. Today we're going to be talking about cells, which are the building blocks of life. Here is a quick agenda for what we're going to be discussing today. I'm going to give a quick introduction, then we're going to talk about cell structure and function, cell types and diversity, cellular processes, cell signaling and communication, and finally end with conclusion and next steps. Basically, I'm going to talk about each topic and then introduce some of the diseases relating to the topic. I love pathology and anything about diseases, so I tried to incorporate as much of it as I could for today. Some of the information might seem basic, but I promise it's important. So first, let's start with a quick introduction. Before we start, it's important to understand why we're learning about cells in the first place and the importance of understanding cells in medicine. So why is learning about cells important in medicine? Cells are the fundamental units of life, so understanding cells is essential for studying biology, diseases, and medicine. Understanding cell structure, function, and communication is key to diagnosing, treating, and preventing diseases. You need to know what normal is in order to identify when there are diseases and abnormalities. Most diseases originate at the cellular level, such as cancer, which is uncontrolled cell division, or Alzheimer's disease, which is a brain disease that destroys memory and important mental functions. Next, genetic disorders result from mutations at the cellular level, and therapies like gene editing directly target these cellular changes. For example, sickle cell anemia is an inherited blood disorder that causes red blood cells to become abnormally shaped, leading to a shortage of healthy red blood cells. Also, medicines often target specific cellular processes or structures. For example, antibodies target bacterial cell walls, while chemotherapy drugs inhibit cancer cell division. Furthermore, knowing how immune cells function and communicate is important in developing vaccines, therapies, and treatments. Finally, cell signaling is crucial in maintaining homeostasis. Dysfunction in cell communication can lead to chronic diseases such as diabetes. As I said earlier, it's important to learn about cells, their structure, and their functions so you know what normal is in order to identify when there are diseases and abnormalities. So many diseases and genetic disorders occur at the cellular level, and medicines often target specific cellular processes or structures. So that's why it's important to learn about cells, their structures, and their functions. So now we're going to talk about cell structure and function. For anyone who took bio in school, this might seem like basic information and things you've already learned, but I promise once we establish the basics and foundation, we can go into the other more interesting medical topics. So first, let's talk about the parts of a cell. There are 3 parts of a cell: the cell membrane, nucleus, and the cytoplasm, where all the organelles are. The cell membrane is a semi-permeable barrier that controls and regulates what enters and exits the cell. It separates/protects the interior of the cell from the outside environment. Next, we have the nucleus. The nucleus is the control center of the cell and it contains the genetic material of the cell, or the DNA. Finally, the cytoplasm, which is a gel-like substance that fills the cell. This is where the organelles are suspended. The organelles within the cytoplasm each perform specialized functions essential to the cell's survival and operation. So now we're going to quickly go over the main organelles in a human cell. We're going to talk about their functions and then talk about some of the diseases or dysfunction that may occur in these organelles. Make sure to pay attention and take notes if you want, because your assignment for this week will be to create a model/diagram of a human cell and research some of the diseases that may occur at the cellular level. So first we had the nucleus, as we mentioned on the previous slide. slide. The nucleus is the control center of the cell, and it contains the genetic material, or DNA. Next, we have the mitochondria, which is, as I'm sure many of you have heard, it's the powerhouse of the cell. And what this means is that the mitochondria generates energy through ATP. Next, we have ribosomes, which are the sites of protein synthesis. And then we have the endoplasmic reticulum, or ER. And there's two types of endoplasmic reticulum, rough ER and smooth ER. So the difference between the two is that the rough ER is studded with ribosomes and it's involved in protein synthesis, while the smooth ER has no ribosomes on it and it's involved in lipid synthesis and detoxification. The Golgi apparatus processes, packages, and distributes proteins and lipids. Next, we have centrioles, which are involved in cell division. They organize microtubules and the mitotic spindle. Then we have microtubules, which are part of the cytoskeleton. They provide structure and assist with cell movement, transport, and division. Then we have lysosomes, which contain digestive enzymes to break down waste materials and cellular debris. And finally, we have vacuoles, which are storage compartments for water, nutrients, and waste, and they help with waste management. So since we're talking about human cells, we're talking about eukaryotic cells. So that means that instead of one central vacuole, like what there is in plant cells, there are smaller vacuoles, multiple smaller vacuoles in human cells. Okay, so now I'm going to introduce some diseases to you guys that occur in organelles. First, we have cystic fibrosis, which affects the endoplasmic reticulum. Cystic fibrosis is caused by a mutation in the CFTR protein, which is synthesized and processed in the ER. The mutation causes the protein to fold improperly. Cystic fibrosis leads to thick mucus buildup in the organs, especially the lungs, which leads to respiratory and digestive issues. So this picture shows how cystic fibrosis results in an obstruction in the airflow in the lungs, which causes issues. Another example is Tay-Sachs disease, which affects the lysosomes. Tay-Sachs disease is a lysosomal storage disorder where a crucial enzyme, hex A, needed to break down certain lipids within the lysosome is deficient. This leads to a buildup of these fatty substances in the cell due to the lack of lysosomes. And these fatty substances are going to build up, particularly in nerve cells. And this results in nerve cell damage, causing severe neurological impact. The next disease is progeria, which is a rare genetic disorder that affects the nucleus. It's caused by a mutation in the LMNA gene, which produces a protein that holds the nucleus of a cell together. So this mutation affects nuclear stability and results in an unstable nuclear envelope, as you can see in the picture here. This ends up causing rapid aging in children. So the picture shows how progeria affects the nucleus by destabilizing the nuclear envelope, and the other part of the picture shows what the physical characteristics of progeria are. And finally, we have mitochondrial myopathy, which is a disorder that impairs the function of mitochondria. It's caused by defects in mitochondrial DNA, and it leads to muscle weakness and neurological problems because of energy production issues. As we were talking about earlier, the mitochondria produces the energy and the ATP for the cell. So if there's defects in the mitochondria, that means that we have energy production issues. And because there is less energy, this is going to lead to muscle weakness and neurological problems. So here's an image of what mitochondrial myopathy looks like in the mitochondria. This is a normal muscle cell, and then this is a mutated muscle cell. So now we're going to talk about cell types and diversity. There are many different types of cells in the human body, each with a different function. So we're just going to go through some of them and talk about their functions. First, we have blood cells. There are three main types of blood cells: red blood cells, white blood cells, and platelets. Red blood cells carry oxygen around the body. White blood cells are part of the immune system and protect the body against invasion by pathogens and foreign substances, and platelets help blood clot to prevent blood loss after injury. Muscle cells are important for movement and support in the body. There are three types of muscle cells: skeletal, cardiac, and smooth. Skeletal muscle cells control voluntary movement, allowing us to perform actions like walking or lifting objects. Cardiac muscle cells are found only in the heart and allow the heart to contract. Smooth muscle cells are involved in involuntary movements, such as in the digestive tract, where it helps move food throughout the body. And then we have nerve cells or neurons. These cells receive, process, and transmit electrical signals throughout the body. So basically, they receive and send messages from the body to the brain and back to the body. Next, we have epithelial cells that line the surfaces and cavities of organs and structures throughout the body. They serve as a protective barrier and control permeability. Then we have endothelial cells that line blood vessels. They're important in regulating blood flow, blood pressure, and the passage of materials between the bloodstream and surrounding tissues. Next, we have stem cells. Stem cells are able to develop into different specialized cell types in the body depending on need. So they can develop into muscle cells or nerve cells, and they're essential for growth, healing, and repairing tissues. Finally, we have the bone cells. So the osteoblasts build new bone tissue, osteoclasts break down and reabsorb bone tissue, and osteocytes maintain bone tissue and respond to mechanical stress. Next, we have fat cells, which store energy in the form of fat. And then we have sex cells, which are sperm cells in males and egg cells in females. And sex cells are important for reproduction. So these are just a few examples of the different types of cells in the human body. Each of them has a specialized function, and each of them play a role in the human body. So if there's a defect in even one of them, this can cause major issues. So now we're going to talk about some of the diseases that are associated with the cell types. First, we have Parkinson's disease, which affects neurons. Parkinson's disease causes unintended or uncontrollable movements such as shaking, stiffness, and difficulty with balance and coordination. In the substantia nigra in Parkinson's patients, which is this in the picture, dopamine neurons in the pathway degenerate. And because there's degeneration of the neurons that produce dopamine, this leads to the movement issues like shaking, stiffness, and difficulty with balance and coordination. The next example we have is muscular dystrophy, which affects muscle cells. So in muscular dystrophy, there are the genetic mutations that affect proteins necessary for muscle function. So this leads to muscle weakness and the progressive loss of muscle mass because the muscle cells are directly affected and there's mutations in them. And then finally, we have leukemia, which affects white blood cells. The cause of leukemia is cancer affecting white blood cells, and leukemia leads to the overproduction of abnormal cells, which leads to immune deficiencies and organ dysfunction. So these are just 3 examples of diseases that can affect the unique cell types in the human body. This picture shows Parkinson's disease, and then this picture shows the difference between normal and then muscular dystrophy. So now we're going to talk about cellular processes, or specifically the central dogma. So if you took biology in school, you might have heard about the central dogma, but I'm going to quickly go over it anyways. So basically, the central dogma describes the flow of genetic information within a cell from DNA to RNA to protein. It explains the process of the production of proteins, which perform most of the cellular functions in our bodies. The first step is DNA replication, where a cell copies its DNA before it divides. In DNA replication, the DNA double helix unwinds, and each strand serves as a template for a new strand. DNA polymerase reads both strands of DNA to create a copy of each strand, and this results in 2 identical DNA molecules, each with 1 original strand and 1 new strand. This step is important because any error can introduce mutations that may disrupt cellular function or lead to disease, as we'll talk about in the next slide. So after DNA replication, we have transcription, where DNA is transcribed into RNA. During transcription, the information in a DNA sequence, a gene, is copied into messenger RNA, or mRNA. In the nucleus, an enzyme called RNA polymerase binds the DNA and creates a complementary mRNA strand. The mRNA strand carries the genetic code from the DNA in the nucleus to the ribosomes in the cytoplasm. And finally, we have translation, where RNA is translated into a protein. In the cytoplasm, ribosomes read the mRNA sequence in groups of 3 nucleotides called codons. Each codon corresponds to a specific amino acid, and the tRNA molecules bring amino acids to the ribosome. The sequence of amino acids forms a specific protein, which then folds to a shape essential for its function in the cell. So basically, the central dogma just describes how the cell goes from DNA to RNA to protein, and it explains the process of the production of proteins, which perform most of the cellular functions in our bodies. So now we're going to talk about Huntington's disease, which is a disease that impacts the central dogma. So Huntington's disease is caused by a mutation in the HTT gene on chromosome 4, which codes for the protein huntingtin. Specifically, this mutation results from an abnormal repetition of the CAG codon in the HTT gene. And this disease directly impacts the central dogma. Because of the repetition of the DNA sequence repeating the CAG codon, the CAG repeats are transcribed into RNA, which carries on this abnormal sequence. During translation, the abnormal mRNA sequence is then translated into a Huntington protein with an abnormal glutamine sequence. The mutated Huntington protein misfolds and aggregates in neurons, which leads to progressive cell death in specific brain regions. So basically, the mutation started in DNA, and then it carried on all the way from DNA to mRNA to protein. And this results in severe neurological symptoms, including movement disorders and cognitive decline. Diseases like Huntington's often arise because of errors during DNA replication, which is why it's so important to know cellular processes like the central dogma, so you are able to identify when there are diseases and abnormalities happening. For example, Huntington's disease. And then this is just a picture of a normal brain, and then what it looks like when there's Huntington's disease. Okay, so now we're going to move on to cell signaling and communication, which is the final topic of today's lesson. Cell-to-cell communication is critical for the function and survival of cells. So cells communicate through three general ways: direct contact, local signaling, and long-distance signaling. First, we have direct contact. In direct contact, cells communicate directly through cell junctions, specifically gap junctions in human cells. These gap junctions allow signaling substances and other materials to pass freely between adjacent cells. So this image shows an example of direct contact. The gap junction is allowing the cells to signal to one another, bypassing the cell membrane. And each cell is the target of the other, so they're communicating directly to one another. The second way cells communicate is through local signaling. This is where a cell will release chemical messages that travel a short distance through the extracellular fluid until it reaches the target cell. Once it reaches the target cell, the chemical messages will cause a response in the target cell. As you can see in the picture, a ligand is the signaling molecule that is released, and it binds to the receptor on the target cell, which will initiate a response. And then the final way cells communicate is long-distance signaling. Cells will release signaling molecules or hormones into the circulatory system, the bloodstream, in order to reach target cells located far away in the body. Since long-distance signaling utilizes hormones, it's also called endocrine signaling. And as you can see in the picture, the signaling molecules are released into the bloodstream, and they travel in the bloodstream until they reach the target cell. So basically, the three types of communication are pretty self-explanatory through the name. We have direct contact, local signaling, and long-distance signaling. So now we're just going to go over some examples of each type of cell communication. An example of direct contact is heart cells. Heart cells use gap junctions to communicate in order to synchronize contraction. Gap junction failure can contribute to ischemia. During ischemia, gap junctions between cardiac muscle cells close partially, as you can see in the picture. This disruption in communication disturbs the synchronization of the heart contractions, leading to an irregular heartbeat. This irregular heartbeat results in ischemia, where the heart muscle doesn't receive enough oxygen due to reduced blood flow in the heart. So this is just an example of direct contact and a disease that happens if there's a failure in direct contact. So now let's talk about local signaling. An example of local signaling is when a wound occurs, cells at the injury site like platelets and immune cells release growth factors. These growth cells bind to receptors on nearby cells, like fibroblasts and endothelial cells, and this binding activates those cells to produce new tissue, blood vessels, and components necessary for wound closure and tissue regeneration. If this pathway doesn't work, it results in chronic wounds, which heal slowly or not at all. And this can lead to infections or tissue necrosis, which is death of the tissue. And as you can see in this picture, it's just comparing what normal wound healing looks like and then what chronic wound healing looks like. And chronic wound healing happens when there's impairment in the local signaling pathway of growth factors. Okay, so finally, we're going to talk about long distance signaling. So, an example of long distance signaling is in the pancreas. The pancreas releases insulin, a hormone, which travels through the bloodstream to target cells. Insulin helps move glucose from the bloodstream into the cells throughout the body, which allows the body to maintain stable blood glucose levels. However, in diabetes, the insulin signaling pathway is impaired, which leads to difficulties in managing blood sugar. The impairment prevents glucose from being properly absorbed and stored, which leads to persistently high blood sugar. There are two main types of diabetes. Type 1 diabetes occurs when the immune system mistakenly attacks and destroys insulin producing cells in the pancreas, and as a result, little to no insulin is produced, making it difficult for the body to regulate blood sugar. Type 2 diabetes occurs when the body cells become resistant to insulin's effect. This means that glucose is not effectively taken up by the cells and is left circulating in the blood, resulting in high blood sugar levels. So, diabetes is a very, very common disease, and it happens because the long distance signaling pathway doesn't work. And the two types of diabetes are type 1 and type 2, and the difference between the two is that type 1 destroys insulin producing cells in the pancreas, so little to no insulin is produced. And in type 2 diabetes, insulin is still being produced, it's just not working properly. So glucose is not effectively taken up by the cells. So this picture just shows how insulin works. Okay, so that's everything we have for today's lesson. So we're going to quickly talk about the homework assignment for this week. So there's two parts to your homework assignment. Part one is to create a model or diagram of a human cell and label its components. So you can use the picture when we were talking about organelles in the earlier slides as reference. And then part two is to briefly research 3 different diseases that occur at the cellular level. So you can pick any disease that was mentioned in today's presentation, or you can research new diseases that you're personally interested in. So thank you so much for coming to today's lesson. I hope you liked today's lesson and topic. Next week will be about the bones, joints, and skeletal system, and we're going to have our first mentorship session about starting your journey in biology and medical studies. So make sure you don't miss next week's lesson and mentorship session. Thank you so much for coming to today's session or listening to the recording right now. I hope you have a good week. Don't forget to do your homework.